Do you know that sepsis affects over 1.7 million people annually in the U.S.? Do you know that sepsis causes approximately 350,000 deaths each year, making it the third leading cause of hospital deaths in the country? Sepsis is a complex and potentially life-threatening condition that arises from the body's response to an infection. Its mechanism involves a dysregulated immune response that triggers widespread inflammation throughout the body. In sepsis, the immune response becomes dysregulated and disproportionate, leading to an overwhelming release of inflammatory molecules like cytokines, known as a cytokine storm. This excessive inflammation causes widespread inflammation throughout the body, damaging tissues and organs. Common symptoms of sepsis include high or low body temperature, signs of infection, mental decline, and confusion. Patients may report severe symptoms like intense pain or shortness of breath, often mistaking early sepsis symptoms for other serious conditions like heart attacks or strokes, making it challenging for doctors to provide prompt and accurate treatment based on presented symptoms. The treatment of sepsis is a race against time. For every hour delayed in treating sepsis, there's a 9% increase in the risk of death for the patient. Experts say that with timely treatment, up to 80% of sepsis-related deaths can be prevented. Moreover, most sepsis diagnoses currently rely on time-consuming culture-based methods, including antibiotic sensitivity determination, which can take several days to yield results. Even if sepsis patients initially undergo antimicrobial therapy, resistance could lead to mortality rates as high as 50%. Therefore, being able to subtype sepsis in patients at the molecular level would lead to many potential clinical benefits, enabling doctors to choose suitable therapies and treatment settings. This makes rapid and accurate diagnosis crucial for sepsis, where time is of the essence. While sepsis-related deaths decreased from 2000 to 2019, according to CDC data, sepsis-related mortality rates among individuals aged 65 and above increased from 277 per 100,000 in 2019 to 331 per 100,000 in 2021. In addition, sepsis-related hospital costs in the U.S. amount to about $62 billion, surpassing the combined costs of heart attacks and strokes. Such high costs are drawing attention from U.S. healthcare institutions. To enhance sepsis management, in August this year, the CDC introduced a new initiative, launching the Hospital Sepsis Program Core Elements Plan to help hospitals establish procedures aiding in early sepsis identification and treatment. It's a free resource center for hospitals to initiate or improve their facilities' sepsis identification and resolution. Manufacturers in the industry are also finding ways to make sepsis diagnosis faster. On November 28th, Inflammatics, a pioneering molecular diagnostics company, announced that it has received FDA Breakthrough Device designation to the company's lead product, the Tri-Verity Acute Infection and Sepsis Test System. Although still under development, the Tri-Verity test is designed to provide 3 independent readouts that reflect the likelihood of a bacterial infection, the likelihood of a viral infection, and the risk of severe illness. On November 20th, the U.S. National Institute of Allergy and Infectious Diseases signed a 3-year, $5.5 million contract with Siemens Healthineers. This funding assists Siemens in developing next-generation sequencing, NGS technology, to guide antibiotic therapies for sepsis patients. Siemens will develop a technology capable of identifying bacteria and fungi in patients' blood within 6 hours of blood collection, offering some resistance information. 1 week before the collaboration, FDA approved Ad Astra Diagnostics' 510K clearance for their POC blood analyzer, QScout RLD, an analyzer that can detect sepsis. The QScout RLD Rapid Leukocyte Differential instrument screens infections, allergies, and blood-related cancers, including leukemia. It measures immunoglobulins, providing sepsis identification within 24 hours, comparable to lactate and procalcitonin. Early in November, Danaher Corporation has announced its partnership with the University of Oxford, both collaborating to develop a new test to enable precision medicine care for sepsis. The novel test will leverage rapid molecular diagnostic technologies provided by Cepheid, a Danaher subsidiary, to pinpoint different subtypes of sepsis and allow the development of novel personalized care paths, including which targeted therapies are most likely to help. At the same time, Oxford Nanopore, a nanopore sequencing company, collaborated with infectious disease diagnostic firm Day Zero Diagnostics to develop a sepsis diagnostic solution. The collaboration aims to use Day Zero's highly enriched sample preparation technology and AI-driven genome microbial identification and antibiotic sensitivity pipeline, along with Oxford Nanopore's high-throughput Promethion 2 solo sequencer, for same-day identification and genomic-based antibiotic susceptibility analysis without blood culturing. The capital market also favors rapid sepsis diagnosis. November 15th, Cytovale announced the completion of its Series C financing, raising $84 million to support the commercialization of Intelisep, Cytovale's 10-minute sepsis diagnostic product. The product received FDA 510(k) clearance in December last year. Cytovale's Intelisep system extracts tens of thousands of cells from a blood sample and stimulates them by applying physical pressure. The response to cell compression differs between infectious and non-infectious patients, and these patterns are acquired through machine learning programs. Despite several other sepsis identification or infection control tests available for inpatient and emergency care environments, Intelisep is the only test focusing on measuring cell dysregulation in host responses and approved for use in emergency departments. Apart from Cytovale's Intelisep, another expert in sepsis diagnostics is T2 Biosystems, which has received FDA clearance for its products. T2 Biosystems has introduced the T2Dx instrument, T2 Biothreat Panel, T2 Bacteria Panel, and T2 Candida Panel. These panels from T2 can identify pathogens directly from whole blood samples within 3 to 5 hours. With so many developments in sepsis diagnosis during November, it's evident that U.S. society is increasingly valuing rapid sepsis diagnosis. The sepsis testing market is heating up, and the focus is on speed and comprehensive coverage. The faster the better seems to be an unchanging truth.